to bring our students to for a lot of different types of activities, including hands-on labs and research opportunities. And they have a citizen science program where they collect marine life inventory data regularly, monthly. They have other various interns coming in from time to time, collecting different kinds of data and working on information. And so I just wanted to share a little bit about that and kind of talk to you about the relevance of citizen science and student research. So let's see, I can share my screen, is that right?
Yes, feel free to go ahead.
Okay, so let me just make sure that I've got what I want here. Here we go. All right, so once again, you would think after doing this all year, I wouldn't be clicking on a million things, but I still am. Okay, here we go. All right. Okay, so again, I am a professor of organismal biology and marine science at Coastline Community College. I got my training in ecology at Syracuse University, so way back on the East Coast. And I actually worked in terrestrial systems, and I'll talk just a little bit about that today. I'm going to torture you a little bit, well, kind of a lot with some math, so that we can look at some of the statistical analyses we've been applying to data that citizen scientists like you have been collecting. And also to give you a little bit more about my background, so that if I say name of a plankton wrong, or if I've spelled it wrong, you'll know that I'm actually a terrestrial ecologist that has transitioned kind of recently into marine science. And so we're going to defer to Joanna as the expert on the real details that we'll be looking at today. But I've been able to apply a lot of the similar statistical analyses approaches to the types of data that you all have been collecting. And so I'd like to share some of the results of that with you, again, with the objective of just showing you what kind of relevance this type of data has for the scientific community. So one of the first things that the students that I work with and I approached this group of individuals with was just looking to see if we could work with the data they had been collecting over several years now, and see if the way that they were collecting it reflected patterns that we would see, that we would expect to see if it had been a very sophisticated scientific lab collecting that kind of data, right? Is there relevance to the way that a citizen scientist group collects their data in such a way that we could actually use it to explain the communities that they've been sampling in? As you know, you guys do this on a regular basis. You do it more frequently than regular scientists in that area probably do it. And your resources are a little different than ours. We have to search for funding, and we have a much bigger kind of set of hoops to go through in order to be able to collect data in the way that you guys collect it. And so wouldn't it be great if we could actually really start using that to explain our communities? And this is happening in many places across the nation and the world now. So we wanted to just see how close are we to really capturing the community by collecting through the citizen science programs. And we selected the one at Back Bay. It's very close to our campus. And so our second objective was to provide our students some hands-on learning opportunities. So by working with the citizen scientist programs at Newport Back Bay Science Center, our students in not only our non-majors marine science classes, but also in our majors biology classes, were able to get experience in collecting data. So going out into the field, using different tools, experiencing what it's like to, no matter what the climate is, no matter what the weather is that day or that time of year, your job is to go out and collect this data, how to do it, how to enter it and manage a data set. I'll point out some areas where that proves to be very problematic and was really a lot of work for the students, more than they actually anticipated. And so those are really important skills for students that want to pursue a career in science. And then for non-majors, it's also just a really valuable way for them to engage with science to begin with, and also to see things that are in their backyard, right? Really get their hands in the water, literally, and see these organisms and touch them and measure them and identify them. So that was one of the values that we find in doing these kinds of things with students. We also wanted to provide, and we want to continue to provide, opportunities for students to build relationships with scientists that are in our community. And then, of course, we get to the trickier steps where we are looking at data analysis, right? So really applying statistical analyses to these data sets that they've collected themselves, entered, managed from previously collected data as well, and then put that together in something and then speak about it in front of people, which again, on any topic, presenting is, I think, like one of the top fears of most humans, right? And so that's a valuable skill no matter what the career pathway is for our students. Our last objective is to communicate with the public, right? So to get the word out about these programs that are going on. I heard Joanna saying, you know, it's hard to get people to participate and attend, and I agree. That'll be one of the things that we talk about when we talk about citizen science in general. And so the more that we can do things like this, the more we share the work that people in our community are doing with the rest of our community. So these were our main objectives. So I am going to torture you with some math and some statistics, but our objective is not to explain the community right now. Our objective is to see, can this data be used? And can we provide opportunities for our students and interact with our community through these programs? So just a little bit of background about where we were working. So here is a really cool view of the Back Bay, Newport Back Bay. So you can see Irvine back in the distance. You can see the Santa Ana Mountains. This is a part of the Santa Ana watershed. So it is where we're particularly located there. It's a little different from I believe you guys are connected directly to the Santa Ana River, whereas this water is coming from the San Diego Creek. So it's a little bit of a different part of the watershed, but again, we're all part of this same watershed that's ultimately originating in the San Bernardino Mountains. Again, back here in Back Bay, we have most of this coming a bit more locally and actually originating within Orange County itself. Again, the program at Back Bay Science Center is designed to work with local students of all ages. I believe their youngest age, they like to have, I think, 12 and above is what they tend to work with. But again, we're talking about middle school, high school, and college. And then, of course, citizens in the community. And they are partnered with California Department of Fish and Wildlife, the City of Newport, UCI, and the Orange Healthcare Agency. So this is where we're talking about here. You guys can see my cursor, right? If not, give me a shout. So here's the Back Bay. Here's Upper Newport Back Bay. So this is going to be more of our harbor area, right? And then back here is where we're working in the estuary. And then if we continue back, we're going to go under the toll road, and you can see San Diego Creek is the main source of the water, with some also coming from Peters Canyon Wash as well. And as you know, a lot of this area, a lot of these waterways have been paved. And so what used to trickle through this watershed is now kind of more like flushing a toilet. When we get a rain, right, we just get this big, huge amount of runoff coming through the county, through mostly developed areas. And then we have this little windy place here where hopefully some of that can interact with the estuary there, and we can get a little bit of filtration, a little bit of absorption of some of the extra nutrients that might be running off. And we're supporting a very diverse community of organisms, from birds to fish to, as we'll see, obviously microbes like plankton that are supporting that food web there. And that water coming through then, of course, is going to carry a myriad of different things washing off of our developed areas. If we have a little bit of residence time there, even though we've had a lot of paved waterways that lead up to that, we do get some ecosystem services, just as you guys are used to talking about and thinking about up in Bolsa, Chica, right, where we have plants that are adapted to extreme conditions so they can absorb some of the toxins coming in. We have nurseries for fish and birds. And so this is a very highly valuable type of a resource for our county. And it's one of the few like Bolsa Chica that remains anymore. So this is where the marine life inventory is collected monthly by the Back Bay Science staff. And the staff opens that to the community to participate in the collection. So the students, again, high school, middle school, college, mostly non-majors, some trained staff, of course, but really a very large group of the community, completely untrained until they get there, are collecting water quality information. They're doing plankton pulls, and they are capturing and identifying, and of course releasing again the marine organisms that are sampled at that time. They have volunteers that enter the data into their database. And then we've recently started to work with some of these spreadsheets and again, see what this information might be able to tell experts about the community in general. So again, our purpose for this talk is to consider whether or not this data collected by local untrained citizen scientists, or at least trained within a very short amount of time, could this be relevant to ecological studies? Are these things we could actually publish? And to be honest with you, as a scientist and watching how the students enter the data and knowing how my students work with data, I would support the null hypothesis. I would anticipate the null hypothesis of, yeah, there's going to be too much variation for us to be able to use this data. The alternative, of course, is that the data that's collected by citizen science can be used to facilitate the understanding of these systems. Now, again, remember, we also wanted to provide students learning opportunities. And so the second perspective that we're looking at in this talk is, what are the students experiencing? What are they able to gain from these programs and participation with the citizen scientists? And so I left this research question and hypothesis as the most recent student who worked with this data and presented it had entered into her presentation. And I will share that she gave this presentation at UCLA. They had an opportunity for student researchers to present, very much like a scientist would present at a conference. It was a fantastic opportunity for the students. And so again, we checked that box for sure. And this was her question. She came up with this on her own. I didn't edit this. I might have edited the hypothesis just a little bit, but I pretty much left it there so you could see what she came up with. And so she wanted to know, is species abundance related to environmental characteristics here at Back Bay? And her hypothesis was that the species abundance, as she has written, can be influenced by a variable of factors. I guess I didn't edit it that well. For example, salinity, pH, temperature. And so she's predicting that the availability of nitrogen and phosphorus will influence the species abundance as reflected in the marine life inventory collections. And this was another really valuable thing, I thought, for the students, this relevance piece. So I also left this in from her presentation. And she came up with these points of why this matters. Why is this even important to us? Why do we care? And to me, that's one of the biggest things that we can give our students, right? Is why are you doing this? Why are you doing all these manipulations with numbers and going out into the field? Who cares? And this is especially valuable with our non-majors, students that are going into very different fields, but kind of have to do this science work. You know, why does this matter? Why is this important? And so she came up with these 3 points, among others. And many of you, as you're appear to be dedicated citizen scientists, and you're involved with Bolsa Chica as well. So you probably already know this, but just to go through kind of what she found, and in case maybe you haven't considered this, marine animals, of course, have been negatively impacted by human activities. And so she thinks we need to monitor what's going on. She had, upon reading the article that this chart on the right-hand side comes from, percent decline from historical baseline, she found that the known diversity, so what's published, really only represents a fraction of what we actually know about the diversity, the biodiversity in the ocean. And so we still have about 95% estimated that is unknown. But from what we do know, as you can see in the chart that she included there, we have almost, let's see, at least 90% reduction from historical baseline in all of the groups of marine animals that we have records for. And so that bar there shows how many records we have for each of these groups, again, from historical things that were collected from fishermen and previous marine scientists. And you can see that for the majority of them, there's only the large pelagics and the whales. And the whales, again, you can see that difference between that gray bar and that white bar. So the white bar was, so it's like 82% of the population was reduced over the history that we have measured them. And then where that gray bar starts is what we've been able to accomplish in the last few years. So that's the change. So we've dropped, now they're only about 68% less whales. It doesn't sound as encouraging when you think of the actual numbers, but when you look at the amount that our conservation efforts have kind of cut that number almost in half, and helped to restore some of the population of our whales. It's a little more encouraging, similarly for our pinnipeds, otters, and others. But for the majority of our populations, as you can see, the decline in their numbers is just phenomenal. And again, these are human impacts that have resulted in these declines. And her last point is there have been many large-scale approaches, but we still have a long way to go to know really what these values actually are, and really even what is out there. So that was the relevance she found in doing her project. So the data that she's working with and that we'll be looking at in a few minutes comes from a couple of different types of collections. They do a bottom trawl monthly along with what's called a beach scene. And so you can see the top picture there is representative of what a bottom trawl looks like. And then the beach scene is in the bottom picture. And so ultimately we have these very large nets that are pulled behind a boat, and we collect either along the bottom or kind of in that upper and middle region near the shore. We bring those organisms in and we identify them, we count them, we measure them. We also do a plankton drag. So you can see there the bullet point, our plankton data was collected. And I may be wrong on the size of our net. It's anywhere from 100 to 150 microns. It's much like what you use at Bolsa Chica, Amigos to Bolsa Chica. And then we have our little sample bottle at the end. And then unfortunately, very unfortunately, what they did was they attempted to preserve their samples in solution, and they did not get the preservation mixture right. And so we actually had 0 results for plankton from their marine life inventory data that they had collected over the last, I'll say at least 10 years, right? So they have the larger organisms, they have the benthic organisms, they have or the pelagic organisms, but those microbes at the top, our plankton, are missing from their data. And that's kind of one of those points that we'll kind of start to consider when we think about what we can do to support you as citizen scientists in some of these methods. So this is what a beach scene looks like. And so you can see the students and the staff there are involved in pulling that very large net into shore. You've got volunteers on the boat. I think I was actually driving that day. Yeah, it was super fun. And I want to just show you a couple of other pictures. I'm going to switch to some Google photos here just to show you the kinds of experiences and the kinds of things that students are seeing from these samples. So this is from our bottom trawl. You can see all that great, all the chlorophyta and our sponges, and I'm forgetting the name of our, what's that called? The moss. It's like a moss, bryophytes. There we go. No, not bryophytes, bryozoans. There we go, bryozoans. They've got resident larger animals that the students can interact with in addition to collecting data. And so here's the intern that actually committed to about the last, I think we've got a good 10 months, somewhere between 8 and 10 months of data that she committed to collect weekly from, I believe we started in the summer and we ended when our quarantine started, unfortunately. So we've got some catching up to do, and we've got about a year of interrupted data collection. But this was one of our students who signed up for a class that is really just an internship and research class. It's our work-based learning program. And so she was interested in learning how to accomplish a project that involved an experimental design, developing a research question, testing a hypothesis, collecting the data, analyzing the data, and she still has the goal to publish this data. And so she did the majority of the plankton work, since the plankton work has kind of been an area that has needed some attention in the MLI collection. So we will get to look at some plankton data today, in addition to what we were able to use from the MLI information, the inventories that had been done over several years, showing the larger animals. Okay, so here's all the things, the details of how we accomplished not only the collection of the information, but also the analyses that we applied to the data that was collected. And again, we have kind of 2 rounds of collection approaches. So we have the MLI data, which is a monthly program. Again, citizen scientists working here, students working here from all different backgrounds, all different ages. And they are weekly, as you can see in the top bullet point, collecting salinity, temperature, pH, dissolved oxygen. They're using probes. We have a Secchi dish. Disc. I'm sorry if I say that wrong. I could never say it right. And then the other kind of version of our data collection was accomplished by the intern at Coastline. And so this was a biology major with the goal to go on as a scientist. Doing this work, she started to think, maybe I want to be a marine scientist. Her other idea was to work in the medical field as a physician. So I kind of made her question which direction she wanted to go, because she had such a blast doing this. And again, I think really benefited from hands-on work with not only samples, but also the research. So she collected using the Plankton net, our plankton samples that we'll get to see. And we used counting chambers and gridded slides. And that's the best plankton data we've been able to collect so far. That being said, you'll see all the stars, right? And so the stars are all of our areas of concern. So when we go to use this data, there's often a lot of things missing. For example, I didn't have my own Secchi disk for a while. And so there's places where we weren't able to collect visibility information. The plankton information from all of those years of MLI data was missing because the preservation wasn't done properly. This third bullet point here, just entering the data into an Excel file, is a whole other project. As I was even just going through today, I found some typos that made replicates of some of the sample dates. And so it's a lot to really keep track of. And there are areas where if the data is able to be used for, or if we want it to be able to be used for any kind of actual statistical analyses and explanation of the natural community there, we'd have to pay attention to those steps. And again, just putting that information together as an Excel file is, it's a whole protocol. And it's a whole level of editing and a lot of detail to keep track of. So as we go through this data, that's also why I've started these. These would be areas that would present a lot of variability to any data that we try to analyze, right? So there's a lot of room for error there. So now we get to some of the torturous mathematics. Are there mathematicians on, or statisticians participating today? Anyone? You can raise your hand. It's kind of good and kind of bad. No math. Uh-oh, I got Annie. Annie's one. Okay. Well, if Annie's the only one, my chances of getting called out on math mistakes are very, very low, but she may also be the only one not bothered by how much math I'm about to take you through. But please jump in, Annie, if there's a better explanation or another way to think of this. But what we did is we took, now you can imagine if we're looking at organisms from the top of the water column, the middle of the water column, the bottom, the benthos, and then also microbes, that's a lot of information. That's a lot of counts. That's a lot of organisms. And so when you put that into a big Excel file, it looks like what we call a matrix. And it is, it's a matrix. And so we apply some what we call multivariate analyses, right? So we've got a lot of environmental information and we've got a lot of organismal information. And that's a lot for a human brain to put into one kind of a representative value. So we use this thing called multidimensional scaling to analyze the composition of all of those samples. Then we apply a step called the principal component analysis. And so that tells us about our environment, kind of in all the variability, in the way that we have reduced that huge group of numbers into one representative value. How then do we determine what within that environmental value is really making today's sample different from last week's sample or next week's sample, right? Is it the temperature? Was the temperature different? Was the salinity different? Was the visibility different that day? And so applying principal component analysis to this big matrix that we've used multivariate statistical analysis on can give us some of that information. And then that last line there describes how we look at again, when we've taken this big group of numbers that represents a sample of organisms on each day, how do we take that and put it into one value and then compare that value to next week's sample, last week's sample, and the composition that's there? And so we use a technique called the Bray similarity analysis. And so that can show us, was today's sample very similar to last week's sample, or was it very, very different? Was today's summer sample very, very different from winter sample, or was it very, very similar? And so again, that's what the Bray-Curtis similarity can tell us. And so you can see at the very bottom there, it talks about Euclidean distance. So we're going to picture all of these values as placed on a graph, right? So we have our y-axis and we have our x-axis, and Euclidean distance is measuring the difference between 2 points on that graph. And that's how we determine whether these data points are similar or more different. So let's think about a matrix for a moment. So in math class, we all had to go through these kinds of exercises where we took these two groups of numbers. Most of us never knew why, but that's what the math teacher told us to do. So we did it, and it wasn't that difficult, right? When we multiplied two matrices together, or added them together, or subtracted them from each other, it really wasn't that hard of a process. So if you were like me in math, and I was never that great at math, I did my homework, no big deal. These are matrices, they're groups of number, and that's really all I knew or understood. As I went into more advanced math, we start to learn that these matrices can represent a lot of different things, right? So when we solve the interaction of two matrices with each other, we get values that can be mapped on a graph. And so we use this in computer programming, we use this in engineering, we use this in statistical analysis. And so if we have all this information about many different types of environmental variables, and then we have all this information about all of the numbers of organisms we collected on all of these different days, those are two matrices. And so we can start to look at the properties within the matrices, just like we were taught how to do in math class, right? We can do things with them, we can interact with them. And since we can do that, we can also turn those big blocks of numbers into a single point that we could put on a graph. And so now we can start to say, all right, I know there's a lot going on in this sample today, but we just want to know, how similar is everything that's happening on this day to everything that's happening on another day? And so we can apply our statistical analyses to give each of those groups of numbers, each of those matrices, a value. And we can evaluate the differences between their values that way, and we can graph them. And so when you accomplish this, and now, Annie, I'm reaching into something that I'm probably not going to explain so great, so let me know if you have anything to add. But when you start to manipulate matrices and you start to add them together or subtract them or multiply them, especially when you multiply them, you get this thing called a vector, right? You get an eigenvector. And so we'll be looking at using these tools to give meaning to these huge groups of numbers that we've collected, and we want to understand in terms of how it represents the natural community. Okay, so here's your first round of mathematical torture. And so this is where I first learned how to do this with large amounts of community data. Again, remember, I'm coming from a terrestrial ecosystem. So I'm here in Yellowstone National Park collecting data on these little tiny microbes that live in the soil. These microbes are fungi that associate with plants. They're called mycorrhizae. And in a lot of ways, the way that we collect them and condense them and put them on a microscope slide is very, very similar to what we do with plankton. And so then also identifying them, quantifying them, and describing their community at a given place and at a given time is again very similar to what we are doing with the plankton studies at the Back Bay. And so you can see there's triangles and circles on here representing our data points. You can see we have 2 axes, and you can see all these strange names and these strange letters and some vectors. So let's start up at the top here. These names are the names of the plants that we sampled, that these little microbes were associated with. And then each of these numbers here represents a place along the landscape where we sampled them. So we have, for example, here at Stevens Creek, we sampled some plants called Pseudomonas, it's a grass, and we captured what the organisms were in Pseudomonas. And this little triangle here represents an area that we had fenced off from elk and bison and pronghorn deer, these large animals that were coming through and grazing like big lawnmowers. So we left an area, like represented by this triangle here, that was not touched by the grazers. And then we had an area that was just open for the organisms to graze, right? For the large mammals to graze. And that's represented by the circle here. And so by using the techniques that we just talked about, we took all of the critters that we found associated at Stevens Creek with the Pseudomonas, and we were able to represent what that community looks like in values of numbers with 1 point. And that 1 point is represented by this triangle. Remember, this is our ungrazed grasses. And so then we were able to also do the same for our grazed grasses. And when we mapped them, we could find then that, remember, we're looking for similar versus dissimilar. And so the distance between these organisms, when you place those values on a graph, and you can calculate that just like you calculate in geometry, right? The distance between this point on the graph and the distance between that point on the graph. And you can determine how similar or dissimilar the communities are from each other. So we got some really great values here. We see that in every site, our grazed community is very different from our ungrazed. We had shifts in this community. This was a relatively small shift. These were larger shifts, right? Small shift, larger shift. And what's even cooler is, again, as close as you are to each other represents how similar your community is to the neighboring data point. And if you're far away from each other, then you're very different from each other in terms of your community composition. So not only did we get some remarkable differences and statistically significant differences between our grazed and our ungrazed communities, we saw them clustering by the type of plant that they were associated with. And then we got super fancy and we overlaid on top what the environment was doing. And so we found that this varied along a moisture gradient, and that phosphorus was another really important driver of how far apart these types of communities were from each other. So we had grazing impacting our community, we had the identity of the plant impacting our community, and we had what was going on in the community determining and driving our community differences. And so we were able to take those huge data sets and put them into these representative points, analyze them, and find these patterns in what would have been just a huge bunch of numbers, right? So we applied a similar thing to what had been collected by the marine life inventory data. And so this first set of data points represents, it looks like all of our points here, these values are dates. So this would be April 09, February 09, November 09, June 2010. And so the reason we selected just late well, it looks like the majority of '09 and some of 2010. The reason we selected those dates, because that was the most complete set of information we had. And some of the other spreadsheets, some of the other years had a lot of things missing, right? Maybe there were environmental values missing. Maybe there were big places in the data sets that had a lot of zeros. So we really couldn't use that information. And so with this set, these months, the data just happened to be collected in such a way that it was complete enough for us to take what was happening in the water during all those dates and compare it to what was happening with our larger organisms. Remember, the plankton got messed up, so we didn't have any plankton data, but we have our fish and our invertebrate data. And so this is, again, very similar to what we applied to our terrestrial system. We're going to do the same thing. Now, I don't have the axes here. I used a little different program, so it's a little bit different. But we have the same idea of if your symbol is close by another symbol, these are the most similar. These groups, these days that we collected and measured all the animals there, that group of animals was the most similar, for example, between December and the following February than the previous January and that June of 2010, right? So these guys that are far away from everybody had very different community composition on those dates that we collected. The guys that are most closely plotted here are most similar. And we actually get values for that. These two dates, the communities were 85% similar. This date was 35% similar to the rest of all of the other days, right? So that community only shared 35% of the same species that we sampled on other days. And even more different from all the other sample dates was June 2010, right? And that was like 25%, like any of the other communities that we had measured. Okay, so, all right, woo, great. We got some values. Remember, we're evaluating, does this mean anything? Like, can we use this for anything? Well, what's super cool is when we look at what's happening in the environment that day. So again, we apply a similar set of steps that say, okay, was the environment similar on sampling date 1 compared to sampling date 2, or was it very different? And our patterns match. So these 2 days where our community composition was very, very similar, our environmental situation, all of our variables in the environment were very similar. So again, this graph here shows this guy is like this outlier, right? He's very, very different from all the relationships that are shared. We get closer and closer relationships if we have this kind of the end of the fork here in all of these branches would be the most similar to each other. And even if you just give it a quick look, okay, you would think probably May and June would be pretty similar in terms of the water quality, the water conditions. You would expect the winter conditions to be very similar. Something's going on in January of '09 that apparently was very, very different than the rest of all of these dates. And so again, this gives us our first check. Actually, the way all of this was gathered, all these students working together, all these different citizen scientists and some trained staff, what they collected showed real patterns that made sense, patterns that showed our community is similar, very, very similar. In fact, the most similar, the days that the environment is most similar. So again, that was the first slide. There goes the null hypothesis. I cannot accept my null that the data is useless. It's not. It's showing what we would expect to see. And so it's really encouraging. It's really exciting. And I did very little transformation of data. I didn't do anything fancy or massage it or anything like that. This is really what came in from all the students and all the citizen scientists that collected it. So that's really, really, really cool. Now, again, the purpose of the talk isn't to go in and pick apart what was different on January 09 compared to what was happening in December and February. The idea is to see, are we seeing patterns that make any kind of sense, right? Was the data collected valuable? So now I'm going to torture you with just a little bit more math. And this is the principal component analysis. So this means, I lied a little bit. I said we weren't going to get into the nitty gritty about what was so different, which we aren't, but we're going to get some more clues about what was making these days different from each other or more similar to each other. What was it in the environment that was really driving the similarities between our December and our February date and the very, very different things that we saw like in August and January of other years. And so when we apply our statistical analysis to just what's happening in the environment, we find that when we take that spread of numbers in that matrix and we put that onto a representative graph, it starts to get a little crazy here, because with these analyses methods, you're not limited to 2 axes. You can actually have multiple axes. So this is kind of like a 3-dimensional type of a representation of the data, which is really cool because that's more realistic, right? We're not going to be able to really take everything and just slap it onto 2 axes. That's going to leave out a lot of information. And so what our principal component analysis does, it allows that matrix to be kind of this 3-dimensional cloud of numbers in space with a lot of different axes. And then it starts to break it down, and it says, okay, if we take away this axis, how much of the differences between the environments on all those dates can we explain? If we take away this axis, how much can we explain? And so we found that you can explain 60% of the variability in the environment on all those dates with just 3 axes, so X, Y, and Z. And then we're going to look at, okay, of our 3 axes, what is it in those values that's really making one day different from another or similar? And look at what comes out. Salinity and temperature explain the majority of the variability on one axis, and ammonia explained the majority of the variability on another axis. And then we also have a strong role for dissolved oxygen. Now, I'm very encouraged by this, the salinity and the temperature, because we teach all of our brand new, from the very beginning, marine scientists, that this is what drives the majority of the patterns in the ocean, right? So this makes sense. Yes, salinity and temperature should explain most of the patterns that we're seeing in the environment. I'm less confident with these values because our tools to measure them were not fantastic. But it, again, still makes sense. The nitrogen that might be present in the water and the oxygen could certainly be secondary factors that contribute to whatever it is going on in the environment that's different from one day to the next, resulting in a different group of organisms that are successful there. And that's what's trickier about marine science too, is these organisms can move, right? This is a very fluid environment. On land, everything's kind of stuck in one place unless a big rain comes. And so there's more room for organisms, theoretically, to leave an environment if the conditions do not support their needs, right? So it would make sense for marine organisms to be found in a group with an identity that represents their needs in terms of temperature of the water, salinity of the water, and what nutrients are available. And so it's very encouraging. Again, the take-home point is we got meaning to this data that was not collected with a purpose to test any hypotheses at all. We just have students and volunteers out there collecting these critters, counting them, identifying them, measuring them, and it meant something, right? We showed patterns of similarity and differences in our community composition that reflected similarities and differences in the environment. And the drivers of those differences were what we expected. The majority of what was explained was salinity and temperature, kind of hand in hand there, right? That was only on one axis. Be real nice if ammonia really was what was driving the remaining, explaining the remaining variability in those environmental factors each day. But again, I'm not confident because our sampling methods weren't that great. Okay, so let's look at plankton. Again, this is using a similar program, but they've updated a little bit, so it looks a little bit different from our first set of graphs, but we have a similar idea where we have, if you're grouped closer together, you're more similar. If you're laying further apart, if your data points are further apart from the others, then your community is different. And so you can see our sampling dates for our plankton specifically ranged from November of 2018, and then we got all the way up to March, and that's when everybody's lives changed. So, or was it? Okay, anyway, I'm wondering if I have the years wrong now. It's all a blur. I don't know what year anything happened anymore. That might be offset by year. So again, you can see even just editing these values, getting them correctly entered into the spreadsheet is a challenge. So now that I'm looking at this for the first time, again, I think that our dates are off a year because quarantine started in 2020, right? And that's when we were shut down. So I think our year is off. But our patterns, again, we've got some really nice, clear patterns. We've got the majority of this year is very similar in terms of some of our outliers in different months of different years. And we have some of our strongest clustering here, and I identified that. I'm not going to show you that graph just because, again, we're going to start to run out of time, and I know it's a lot of math and data points here. But the value, this cluster here, these guys are about 75% similar to each other based on the same kind of analyses we looked at previously. I haven't gotten down into the nitty gritty on how closely these guys cluster. Again, you can see there were errors in data entry, in date entry there. So those need to be updated. But right here, we have a nice clustering of about 75% similarity. And then sometimes it's easier to look at things that are very, very different than something else and analyze what's driving those differences. And so we've got these 3 points, which are dissimilar from the rest of our sample dates by about 65%, right? They're only 35% similar to the communities that we found on all the other dates. And so this is what I mean by that multi-dimensional space. And so the name of that analysis is non-parametric multi-dimensional scaling. So we're taking these large matrices, these large groups of numbers, and we're letting them be this cloud in space, but we're assigning each cloud a point based on the manipulations that we did, multiplying matrix 1 by matrix 2, right? What were all the values for this day? What were all the values on the environment for that day? We put those together and we get our point in 3-dimensional space. And so that's what you're seeing in this little cube here. And so the clustering, the closeness, and the dissimilarity is kind of nice to see from that perspective as well, that 3-dimensional perspective. So you can see, yeah, these 3 dates really were very different. than what we found in the rest of our sampling dates. We have this guy kind of inching over there. And wouldn't you know, we found a very same thing to what we found with the large animals. Our 75% similarity is the same kind of similarity that we have in the environment on that day. So again, this is September 06, 19 and October 11th. And those were these guys here. These guys here. Our outliers, 1025, 1120, 310. Oops. We're also outliers here. When we look at what's going on in the environment more closely, we find something that I'm more willing to accept as reliable. We have visibility in the water column driving the majority of the differences in our plankton community. And then here we are again, temperature and salinity, hand in hand, sharing kind of this a covariance, a co-varying value that's determining our or influencing, related to our community composition on those same days. And this is, again, something that makes sense when we think back to what has been demonstrated in loads and loads and loads of data and studies in marine science. We're always taught temperature, salinity first. Certainly visibility is going to influence who's able to succeed in that upper level of that photic level of the water column. And so this makes sense. The data that my student collected and analyzed, and we did do fancy transformations with this data. We did do kind of more sophisticated like corrections and things, and more accurate bookkeeping, if you will. And we got similar patterns that made sense in the same way that those large numbers of larger animals collected by citizen scientists made. So this is super, super encouraging. And again, we're checking that first box. Is the information collected by citizen scientists something that we could use to describe and analyze the community? Absolutely. Yeah, if we can just kind of improve and kind of clean up just a few areas, we can really use this data and explain a lot of what's happening in the community and what's happening in the environment. And so this is the last mathematical graphic piece of torture that I will give you. But again, here's the kinds of things that are happening in the environment that are making these dates so very different from these other outliers. And again, what's nice is no matter which way you look at it, we're getting pretty much the same outliers that are matching. They're very, very different in terms of the environment. They're also very, very different in terms of the organisms that we find there. And we find it for our microbes, and we also find it for our large animals. And so this just summarizes some of those principal components. And this is what we mean by the two axes representing it. So in that three-dimensional cloud that you saw in the cube diagram before, if we smash that down and we just pick 2 dimensions, we pick 1 and 2, we again had our visibility, temperature, and salinity really determining how this spread in differences and similarities is falling out. And again, that makes sense with our published data, and it held up in both types of sampling situations. So I'm willing to accept that things like visibility, temperature, and salinity would be drivers in our environment that would influence variability in the species that we found in that environment. The students were able to sift through this huge torture session I just put you through and identify, why do we care about knowing these values, right? They looked at it and they said, well, yeah, we don't know what's going on in the ocean. It's probably important that we figure it out. And they learned about how to accomplish research. They learned about the opportunities in their environment. They collected data. They did an okay job of managing the database. As you can see, there's plenty of places for errors there. They had to learn these pretty sophisticated ways of looking at data and analyzing them and applying statistics. And we had, this student hasn't presented yet. I had a student present at UCLA, and then the students that are in the majors classes have to present at the end. That's their last project. That's their lab midterm or lab final grade, is to present what they've found from their collection. So we have achieved our objectives in this first round of examining the data that was collected by Back Bay Science Center. We found a lot of surprising and very encouraging relevance to the data, repeatable patterns that were driven by the same variables in both settings. And if we can just cooperate a little bit more and tighten up some of these areas where we're sampling, we've got a lot of really valuable information about our natural communities here in very important places that we can use. And so next steps, if we can work together and tighten up our techniques a little bit, work on organizing the data, the step of taking what the citizen scientists collected on their worksheets and putting that into a spreadsheet had some areas that could be improved so that we can really smooth that process for whoever's working on the analyses. And I know Joanna will support me in this. We need to regularly collect that phytoplankton, especially, of course, we'll get zooplankton as well, but if we can collect that, if we can go out and sieve that every week and really analyze who's there and get the counts, then we can start to really look at what's happening in that very important first level provider of all the food for all of these other levels of the food web in those communities. Here's some references. Back Bay Science Center has a website that shows their resources, their activities that they participate in. We finally have a website for our biology department that can give you more information on some of these opportunities available to students. And there's a couple of citations that I included there as well for the data. And do we have time for questions or?
I think we'll have time for like a couple. If anyone has a question for Tanya, feel free to unmute yourself. Also, first of all, thank you, Tanya.
Welcome. Thanks for being here. I hope I didn't get too overwhelming with the data.
No, it's super interesting to know that, you know, the with like a couple, you know, fine tuning.
Yeah, already you've got patterns that mean something. It's really, really cool. Yeah,
it's exciting.
Very exciting.
Yeah, because we actually do have a. Oh, did I hear someone had a question?
I have a few questions and comments, but I want to let other people first. I'm actually, before other people start logging off, because I don't know if people might have to go, but I wanted to just make sure that we talk about Tanya. I don't know if I missed in the very beginning, but I know other people arrived after me. Tanya, would you mind explaining to us what you do at the community college and how is it that you get to teach all these really cool classes? You mentioned that one of the classes you teach is this research-oriented class.
Right.
The students get to do this. That's not something that most of us are familiar with. So maybe if you could just explain a little bit to those who arrived late like me, who, you know, what you do.
Yeah. Sorry, I glazed over myself because I wanted to torture you with numbers mostly. So we got all that taken care of. Right. So like I said, I did my training on land instead of on sea back in New York. And when I came back to California, I worked at several different places teaching. I worked in consulting, environmental consulting a bit, and even completely other field, neuroscience research. I actually have taught human anatomy for decades now, just because it was a job that paid the bills actually, and then I fell in love with it, so it's kind of another area. But I started working full-time with Coastline Community College. They're the sister school to Golden West and OCC. And they're a kind of a strange setup with their campus. They have multiple campuses. They have a lot of online courses, which has been so useful with this crazy last year because we were already prepared for really most of what we had to do online. And there's a lot of opportunities for students to take classes that way. And then in our science department, we offer kind of two areas that you could focus in. You could go more into health sciences and do like your pre-health, PA preparation, med school preparation, nursing school preparation, physical therapy. We have a lot of students working on those goals. And then we have kind of the more traditional biology. And regardless of which path you're going, as a biology-centered major, you're going to take some biology major courses. And we have two. We have our cell and molecular, and we have biodiversity of organisms. And so I teach the biodiversity of organisms. So that would be like majors biology 2, part 2. And this is where we look at all these kinds of things. We look from plankton, we look from bacteria, actually, all the way through mammals. And so they get to work on this too. This is one of the best ways to teach about protists and plankton, right? Because it's in their backyard. We go out and sample it. And then we're also contributing to the database that we've been collecting there. The research course that we have is something different. It's something that anyone that has some background in science can participate in. We call it work-based learning. And so we kind of have those two different directions. We have opportunities in actually biotech and some clinical work for students that want to go into the health profession. And then we have this more kind of traditional research that we can do. And so we partnered with Back Bay, just again, because they're right down the street. They have this little dock. Honestly, we just go out on the dock and we take our plankton collections there. We use their microscope. So we get it done real quick. And then I had been helping them with their monthly MLIs. And again, that's a really great opportunity for any of the students. And so that's how I get to do all this fun, great stuff outside of just the traditional teaching in the classroom.
Very cool. And do you mind stopping sharing your screen so then we can see everybody?
Oh, sure.
And I would love for people to ask questions. So, and I want to thank you for showing us, you pretty much showed it under the hood, right? You showed us What is it that it takes to actually conduct a scientific study that passes the statistical rigors that we expect. And I think your findings are very exciting to all of us who are interested in citizen science. All of us are involved here, most of us through Amigos, and some people probably through other organizations. But you touched on some very important points that we internally strive to improve on. So, you touched on the methodological, let's call them pitfalls, like potential challenge. You gave the example of how they use the wrong preservatives, right? So
yeah, that was a problem.
Right, and you were talking about years of samples, right, that got collected, and you can see that this is something that probably they had someone who gave them some advice many years ago,
right, and
perhaps that didn't get updated, so that shows us the importance of having scientists working alongside those projects and really, and we've been trying to do this more and more here. Of course, I started the Plankton program because Plankton is my expertise, but the idea was ours to expand, right? The idea was ours to invite other scientists that could bring their expertise to the Bolsa Chica.
Yes.
And utilize these amazing folks who know so much about the Bolsa Chica, who are eager to learn more, and who are willing to be trained and conduct this amazing field work that is so valuable and so hard for scientists to do on their own.
So much. And you guys have done so much. And that's what was so exciting. what you've done is useful. It's hugely useful. And you're not the only citizen science program out there. There's lots of people collecting data like this. And so just like you said, it's really important anywhere we can find scientists partnering with citizen scientist programs and the students. You know, it's a very important dynamic, I think, and extremely valuable, the types of collections that you guys are doing. I mean, you have loads, you have years, months of data that's, even without getting fancy and tightening up the techniques, we got some really cool patterns that will be upheld by statistical analyses. So really cool.
So I want to give people a bit of a context. Unless somebody has a question, just raise your hand. But I know, just for the interest of time, I want to give people a little bit of context on what we're doing with citizen science, and then you can think about what Tanya presented and see how it aligns. Because as she said, the Back Bay Science Center is just down the road. And show of hands of who's been at the Back Bay Science Center. I know a lot of you have. Yeah. All right. So you're familiar with the structure that she's talking about, right? Which we used to use the visitor center across the street as our physical space. So obviously working at the Back Bay Science Center it makes it easier for people who are able to use that facility, but we made it happen. One of the things that they do have is personnel, the boat that Tanya got to drive. That's so cool that you got to drive the boat. But the program, one of the programs that she mentioned, which is the marine life inventory that happens once a month, that is something that we could do once everyone is vaccinated and COVID is under control. We could definitely come as a group. We can schedule and we can talk to, I think Robin Madrid is still the one who's booking this kind of stuff, even though she got promoted. But we could all come as a group, and maybe Tanya, you could come with us that day when we finally return to our in-person operations, and we could come and see what they do. They do the fish collection. They do like a much broader work, and then maybe we can share with them what we do with our phytoplankton. We don't preserve samples. So we made a decision years ago to instead of preserve samples, what we do is we take pictures of the live samples.
I think it's much better that way, yeah.
So we do an inventory. Yeah, we do an inventory, and then what we do for those who might be new, and I'm looking here at Sarah, at Karen. I don't think Karen got involved with Flow, Doris, all these people who might not have gone, and Martin, I don't know if you've ever gone to Flow. But what we do is we send a sample to the state. That's why we can call ourselves citizen scientists, because we are contributing to an actual scientific project, which is run by the state. The state Department of Health has that program. And that's why we can call ourselves citizen scientists, because we are collecting samples and data and providing that to the scientists that work for the state. What we would love to do is to get someone with the knowledge that Tanya has, and this is an invitation right here. That's the hook. I'm throwing the hook in the water here.
Yes.
To go through the data that we have, the additional data that we have been collecting, and Judy, you know what I'm talking about, years worth of data into Excel spreadsheets. And those would definitely be something that a group of students advised by a scientist who understands about statistics could probably take a look and see if there's anything there that is publishable.
Yeah.
So that's
one of the things I noticed, and this is what they do too. They take a sample and they send it. That's where the health department was involved as one of their partners. So remember, it took a dedicated student to identify, quantify, right? So with what you guys do, you're looking for something specific. So your methods address that. You address whether or not you're seeing Pseudonychia or Alexandrium, or I know there's another critter probably cropping up now, right? And that's you're looking for those red flags. We can talk about red flags later, Julia, but if in your, there's a couple of things you could do if you wanted to quantify your community. I don't know if you've done this a little bit more now with the work that you've been doing in research, but even if you take images and save those. You could go back and identify and quantify based on the images. That's a sample,
Right.
right?
Yep.
If you do it the same way every day, if you get some of those counting chambers and as you're going through and you're having fun identifying who, just making a note, how many dictylum did we find?
So we from the beginning, we decided we wouldn't do a quantitative approach. Right. So our work is always being focused on qualitative. It's either presence or absence. And so we also do, it's a semi-quantitative analysis in the sense that we identify the most abundant, the dominating group. We do that.
And that's easier to do statistics on too.
Right. And so the goal here is more to see about the temporal scale. So like if we have this really long timeline, do we see seasonality? Do we see this presence of organisms happening over time around the same time of the year, or matching some specific temperature salinity conditions that we're also measuring? So we do have like a plan when we created the program, but we never had the scientists and the students that would come in to do that second part. So you were all very, very invited.
Yes. And see, like without even having a person like Joanna to guide this other group, their data came up with some really great patterns. And so it's very exciting what you guys are collecting, and the potential that it can have towards ecological studies is really important. It's really, really important. So, yeah.
I get to hear that from other people.
Tanya, sorry, I don't mean to interrupt. I did have one more question that was sent to me in the chat. It was regarding the environmental data. It was, were they considering rain records, tidal records, like tide height, atmospheric data?
No.
As related to environmental data?
No, but that is very valuable. I think it could be really valuable to sample at the same tide level when you're doing it. Sorry, what was the other? Oh, rainfall. And if any of you know where to find good rainfall data, we've tried to go through some of the public records, but that's another ambiguous piece of information that we would love to have that I think could be very valuable. And again, I mean, you could go through the weather even, right, and see when we had high rains versus low rains, but there's really not a reliable, that I've found, and I could of course be wrong, there's not a reliable set of environmental data that covers nitrogen, phosphorus, rain levels collected at the same tides, along with the salinity and pH and temperature. It's really hard to find a consistent data set for all of those variables. And I agree, they're important.
One source for some of that information might be USGS data. They track stations up and down various rivers and water bodies that collect some of that information, and including, I think they collect rainfall data, but if they don't, then NOAA does.
How, right? See, that's, I'm glad you said that, because that's, I don't think it's just me. It's hard to find that stuff, but if they did, can you imagine the watershed information we could add into what's happening in our estuaries? That would be really, really cool. So USGS, and I'm sure they have, I wonder if they would have sampling sites along the coast. That's another thing I was thinking of. I know we have many watch places along the coast, but regularly sampled other places that we could use, I agree, would be very valuable. I'm seeing
remote sensing, so it's collected.
All right, yeah, we'll have to check them. I just clicked on a link with some naturalist data, and there could be that as well. That would be useful. I lost my chat window, but there were some other comments.
And Tanya, I think Gwen is posting a link on the iNaturalist in response to Sarah's question about any other project within iNaturalist used for data at all. Gwen, do you want to talk a little bit about iNaturalist? I know that you've worked with it.
Sure. So who uses iNaturalist? Anybody? So, and you can log anything that's a species, and sometimes dead things they'll take too. So anything you observe, you can log there, and then it sort of creates your own life list of things you've seen. And they take auditory and visual, so pictures or audio. And there are a lot of papers being published from iNaturalist data.
Great.
Yeah, there is a lot. It's great. And, you know, there's certain kinds of things you can do with that kind of data, but it depends, of course, on how it's collected and what the intent was originally. But some people do make projects with the intent of trying to, you know, determine range expansions of different species and, you know, spread or change over time. And so a lot of the bioblitzes, part of what some of the bigger bioblitzes are trying to do is collect data on a large scale to compare over time. And then each year there are different sort of focuses, questions, 4 or 5 things, 4 or 5 species they might want you to focus on. Sometimes the sea stars, like lately the sea stars have been of concern, but there are some certain different ones that seem very sensitive to temperature changes or whatever else is going on that year in the last 5 years. So
and Gwen, can I ask the complementary question to the one you asked? Raise your hand if you've never heard of iNaturalist, if you don't know what it is. Okay, so everyone here is aware of what even if you haven't used, right? So it's something that we considered using when we were talking about that collaboration with UCI to do the plants. Remember the plants and COVID? We had the professor that came and talked about us collecting plants and helping them connect with other presenters. Yeah. So in fact, I want to give you an update on that project. They're trying to do now the second year to compare a, you know, normal traffic, normal pollution condition with the one that the data that they collected last year during COVID. So I will be emailing you all. Nancy, I'm looking at you again for that second round because the UCI professors are looking for it. But iNaturalist was used then, Sarah. And so that's one example in which iNaturalist was not used as a hub to store the data. It was just used as a way to identify the organism. And of course, we were storing the data while we were doing that. But then we took that data set and that was used for a project that was external to iNaturalist. So you can see how some of these apps, some of this technology based tools that we have available for citizen science can be used as the main hub or as a supplement, which is kind of cool. And I want to mention too, there are a number of organizations that do kind of what Tanya did. It's so cool to see you, Tanya, thinking in that way alongside this very important question, which is constantly evaluating the quality of the data that's being produced by citizen scientists. This is very important because it is a mechanism for us to show the relevance of citizen science, right? Like once we establish that it's data that is useful, then we can continue applying for funding. We can continue recruiting volunteers in a way that is evidence-based. It's informed by, you know, these kind of quality checks that Tanya performed here. And so there are a number of associations,
scientists to help them, because it's only to their benefit to have this data available. I mean, that's their currency, right? Their publications are their currency. They're hungry for it. And so if we have all this amazing data, which we do already have, and we're just trying to prove, look, this is valuable information that you guys have been collecting. Once we start to publish that and they're reading the publications, then they're going to say, oh, I want to work with Amigos. I want some of the, you know, like your person from UCI did.
Yeah. And alongside, you know, we have now these sources of funding that are specific for citizen science. And that was something that when we started doing citizen science almost 10 years ago, it was very rare to find any big federal or state agency that would be willing to throw money at citizen science, because it was considered something that was too iffy, that, you know, they didn't know it was really something reliable or not. But now you have NASA, you have the EPA, you have at the state level, several agencies that are excited to fund projects that are citizen science based. We are now in the in a process of expanding for citizen science. Tom, are you still in the call? There's Tom there. So Tom and Gwen and we worked really hard to put together, and Tanya was one of the collaborators that said yes to be a partner in this. We wrote a big project proposal to expand flow. You guys remember Martine that came and gave a talk about putting those buoys inside of the wetland and collecting that data. So we didn't get funding from that one application that we did, but we're continuing the process of applications. And recently, actually, I'm going to let Gwen and Tom break the news, because we have good news related to citizen science. Who wants to tell the group what, the recent success in that front? Tom, do you want to share?
I will, yeah. I've told, some of us already know, but we just got some funds from USC Sea Grant to buy some very nice equipment for testing all sorts of things. Gwen can probably explain what they test better than I. I just know we got a really fancy microscope and a...
Yes! Ooh! All right, I'm coming over.
Yep,